Okay, Chapter 7, Section 10. Uh, the phosphate or pho regulons. Okay, we're going to step away from thinking amino acids. Now we're going to be thinking about phosphorus. Okay, phosphorus, remember, inorganic phosphates, going to be important for DNA, RNA, any of our nucleotides, membranes, phospholipids. Remember, you got to have phosphates there. Energy generation, ATP, cell signals. Remember, a lot of our signals, whether you're talking eukaryote or prokaryote, are going to be based on kinases. So you have to have a lot of ATP so that these signals can work. You know, so inorganic phosphate, PO43, or you'll see it, P sub I. P sub I means inorganic phosphate. When it's in high concentrations, the cells are going to take up as much as possible. But more often than not, it's going to be limiting. Okay, it's going to be in a scarcity, which means that whatever bacteria are present in an environment, remember, no environment has only one type of bacteria there. There's always going to be multiple species. They're all in competition with each other. So the competition means that there's going to have to be, different cellular responses to high levels in the environment and scarcity levels, the limited in the environment. So just like we saw in section 9, there are going to be regulons, pho or phosphate, pho regulons, or pho, whatever. Two-component regulatory system is what we're going to be seeing here. When it, that responds to limited inorganic phosphates in the environment. For this, going to be using the example organism Streptomyces. Okay, Streptomyces is one of our sources of antibiotics. Turns out Mother Nature makes better antibiotics than we can. You've heard of them: streptomycin, streptomyces, streptomycin. Huh, there you go. It's a gram-positive soil microbe. Okay, means that since they're in soils, there's going to be a scarcity quite often of many, many nutrients. Turns out inorganic phosphate's one of them. It's the scarcity of inorganic phosphates that has triggered the evolutionary development of antibiotics. Turns out antibiotics are the weapons these bacteria can use against other species of bacteria in their competition to get at what few nutrients are available in their environment. But producing antibiotics is very energy intensive. So if there is an unlimited supply in the environment, why waste? Why waste all that energy producing antibiotics? Only produce the antibiotics in a time of scarcity when the competition matters the most. Sorry, I had to fix the camera. So, low phosphate is going to trigger the pho regulon. Pho. Sorry, I'm hungry. Haven't eaten dinner. Pho regulon, the phosphate regulon, which means, there's going to be some antibiotic production. Not only antibiotics, a whole other hundreds of other genes, but that's a couple of them. What we see happens is that it's a two-component regulatory system. Okay, think about that. Two-component regulatory system. A regulatory system is a series of checks and balances to say yes, no, if, when, do we need it now, of, metabolic activity, gene expression. The two-component part means that there's going to be two proteins. Generally, one's going to be a receptor, and then there's going to be a cytoplasmic protein. So we have a membrane-bound histidine kinase. Histidine kinase. Kinase means it takes the terminal phosphate off ATP, adds it to something. Well, what it adds it to is a histidine, amino acid histidine residue in its target, its substrate. So we refer to that as Fo-R. And the second part of the component is the cytoplasmic transcription regulator. Remember, we talked regulators in a previous chapter. These are the proteins that will bind to promoter regions to either increase the chances of transcription or interfere and decrease the chances. This is Fo-P. Low environmental inorganic phosphate triggers Fo-R to phosphorylate P. That's where you get Fo-P, phosphorylated protein, phosphorylated Fo-P. Fo-P, once phosphorylated, changes its shape. It now has a high affinity for what are referred to as Fo boxes. Okay, these are specific promoter regions that phosphorylated Fo-P will bind to. Okay, remember, this is going to be regulons, operons, individual genes that need to be expressed when inorganic phosphate is in a limited supply. Fo-P can act with, but is also independent of sigma, so it can go and bind to certain other promoters that aren't Fo boxes, but it does so weakly. It does block RNA polymerases, okay, in certain genes to downregulate transcription of genes responsible for, amino acid synthesis, ribosome synthesis, things like that. So it's going to be working against the genes, the regulons that we just talked about last section, in section 9, that are going to be kicked off because of amino acid limitations. And we look at it here. Here's our two components, Fo-R, Fo-P. Low inorganic phosphate will trigger Fo-R to become active. How's it do it? Don't know, don't care. If I don't know, I don't care, neither should you. Just know that it does. Fo-R will then phosphorylate Fo-P. Fo-P will then go to different regions on the chromosome, increasing antibiotic production, because remember, we're now in competition with other bacterial species, fungi, other microbes, archaea, whatever. And increase expression of the genes that make the transporters. Okay, got to increase the transporters in the membrane to bring in as much inorganic phosphate as possible. And then it's also going to repress genes for nitrogen metabolism, for amino acid synthesis, things like that. We're now looking at this Fo regulon as also part of the pathogenic response. Microbes get into us, causing an infection, get into, a low amino acid, or here, a low inorganic phosphate. You don't have a lot of inorganic phosphates in your interstitial fluids. You don't have a lot of inorganic phosphate in your bloodstream. Well, it's going to trigger certain responses. E. coli 0157H7. Remember, this is the one I've talked about in previous lectures. This is the one that causes the bloody diarrhea because it has the Shiga toxin. Well, Fo regulon there's for about 40 genes. Some of them are going to be, for fitness survivability. Others are going to trigger antibiotic resistance inside of you. So there you go. The antibiotics you take may not work against the bacteria, which is why all hospitals always test for resistance and susceptibility. And then it's also going to be maybe the production of toxins, maybe triggered by phosphorylated Fo-P binding, promoter regions that may not be those Fo boxes. We've studied it in Vibrio cholera and Pseudomonas, and we found that the faux regulon, while not directly controlling, may influence the genes responsible for biofilm formation, toxin production, antimicrobial resistant proteins, penicillinase breaks down penicillin, things like that. So while not directly upregulating these activities, it could be a side effect. The weak binding, which causes a weak upregulation of those, genes.